Hello. So I'm just going to give a brief rundown of DNA to RNA, the replication, transcription, and translation, so you can get a better idea of how it all pieces together. So here's the original cell. As you can see, the DNA lives in the nucleus of the cell, and then there's the ribosome over here. Here's the ribosome, and here's the structure actually is the ribosome. And then the nucleus is the big black ball in the middle, and the DNA lives in the nucleus. Now that's in the first cell. Then the cell divides when, it's forming new cells. The cell will divide into two daughter cells. So here's the original cell right up here with the DNA in the nucleus and the ribosome and all that. And the cell breaks into two. And when the cell breaks into two, that DNA, that double strand of DNA, also breaks into two. And one half of the DNA strand goes into one cell, and the other half of the DNA strand goes into the other cell. So this leaves a daughter cell with only half of the DNA strand. So what it needs to do is it needs to replicate the other half of the DNA strand. Now what it does, the DNA replicates the second half of its DNA strand, the missing half, by matching it up with the base pairs that match. So you got A to T right here, and you got T to A, and you've got C to G and G to C. So it creates a second half of its own self with the complementary base pairs on the other side of it. Now that it has the second DNA strand, I erased the first DNA strand, the original DNA strands, just so we could concentrate on this new DNA strand that was created. So this new DNA strand, it needs to send out information outside of the nucleus. But DNA lives inside the nucleus. It can't leave the nucleus. So what it needs to do is it needs to create like a messenger, to carry the messages from inside the nucleus to outside the nucleus, to the ribosome. So this is called transcription. See this white? It looks like a DNA strand, but actually what it does, it does the exact same thing as it did when the DNA needed to make the other half of itself. But instead of making the other half of itself, this second DNA strand makes another strand with complementary base pairs. See the A to T, the A to U, or the T to A, excuse me, the A to U, the G to C, and the C to G. And this new strand is called mRNA because RNA can leave the nucleus where DNA is locked into the nucleus. So that's why it has to create this mRNA strand. So the mRNA strand is created and it will leave the nucleus and it will go attach itself to the ribosome out here. So here's what happens. See the mRNA strand was right here. It was just made and now it left and it went outside the nucleus and it went and attached itself to the ribosome outside of the nucleus. Now here's a close-up of that mRNA strand, the messenger RNA strand. Remember, it's messenger because it's the messenger that carried the information from inside the nucleus with the DNA to outside the nucleus to the ribosome. So see, here it is attached to the ribosome, and it has the complementary base pairs that was created inside the nucleus that match up to the corresponding base pairs on that DNA. Now, while it's out here, while the mRNA is out here attached to the ribosome, there's all these little floaty things. Do you see these? These orange things. There's all these little floaty things, these structures. And each structure, on one side of the structure, on one end, it has complementary base pairs. See the UAG? This one over here has GCA. You see, so on one side of these structures are complementary base pairs in sets of 3. Don't forget that these are sets of 3. And on the other end of these structures is an amino acid, just one amino acid each structure. Now all of those floating along, you saw how they all had different base pairs, complementary base pairs on them. The one that's supposed to match up to the mRNA, as you can see here, we have the mRNA has A, U, and C. So these complementary base pairs that match up to that mRNA, the U, A, and G, this structure attaches itself to the mRNA. All the other structures float off. So the correct structure comes along and attaches itself to the mRNA, leaving this amino acid sticking out. And these structures that we've been talking about, this orange structure that has this the complementary base pairs on one side and the amino acid sticking out of the other side, this is called tRNA. And the process of this structure coming on up and attaching itself to the mRNA, leaving the amino acid sticking out, that's called translation. Then that's why this is called tRNA. The tRNA does the translating. Because what does it translate to? It translates the base pairs into an amino acid. And these three base pairs that we have on each of the tRNA structures, the UAG, that is called a codon.